Very nice introduction. You forgot to mention I was also the guy early in my career who was pulling Ethernet cables through the ceiling. That's how all good informaticists start, and I highly recommend that you do it. In any event, I'm gonna give you a vision for an NCI Cancer Research Data Commons. I'm going to do this in 4 short steps. I'm going to give a brief background. I'm going to talk about the NCI Genomic Data Commons, our Cancer Genomics Cloud pilots, and then finally talk about our Cancer Research Data Commons. For those of you who have been here all day, you know that Warren Kibbe did give a good bit of the overview, the background this morning. But that was 6 hours ago. It's going to be a test of your short-term memory. I'm not going to spend a lot of time on that. There were 2 major initiatives that are drivers for this vision. The first is the Precision Medicine Initiative, and for the NCI in particular, the PMI Oncology. And the key piece here is that detailed data are critical to creating a learning health system. And the required open slide, although there's no F word on this slide, we do support obviously open science, which means open access, open data, open source software, and we have emphasized that in all of our contracts, and this is being more and more emphasized in grants these days, and data liquidity for the cancer community. But also standardization through terminology, through common data elements and case report forms, and interoperability by exposing existing knowledge through the appropriate use of these ontologies and vocabularies and taxonomies. And then ultimately sustainable models for informatics infrastructure, services, data, metadata, and curation, because if it's not sustainable, then it's probably not worth doing in the first place. The second major initiative is the Beau Biden Cancer Moonshot. And again, here I'm just going to focus on the very last bullet, which one of the key drivers here is enhancing data sharing. And Warren talked about this morning about the Blue Ribbon Panel recommendations. I've highlighted the one that's relevant here, which is the creation of a national cancer data ecosystem for sharing and analysis. So this is the second major driver for our vision. So the vision is really to create a cancer research data commons. Don't confuse that with that data ecosystem. This is one small part of that data ecosystem. The goals are to make research data available for discovery and validation of new therapies, maximize the impact and reuse and reproducibility of cancer research. And this is a really important one, changing the incentives for data sharing. This is not a solved problem. But we're all thinking very hard about how we move there. And this, as I said, this is just a key component of a learning national cancer data ecosystem. So with that, I want to talk very briefly about the first foray we've made into this space, which is the Genomic Data Commons. The key data set, the driver for creating the Genomic Data Commons was the TCGA data, the Cancer Genome Atlas, which generated 2.5 petabytes of data from 33 different tumor types, including 10 very rare cancers from over 11,000 patients. It had 7 different data types from 17 different experimental strategies, everything from RNA-seq to whole genome, whole exome, mRNA-seq, genotype array, expression array. So it was a lot of complex data, and it's produced a lot of new knowledge. I don't even know, there have been hundreds of papers that have been published using the TCGA data. It's really helped improve our understanding of the genomic underpinnings of cancer. It's revolutionized how cancer is classified from organ-based to more genetically based, and through identifying genomic characteristics of tumors that can be targeted with existing drugs or help to develop with new drugs. The TCGA, the GDC launched June 6th of 2016, just a little over a year ago, and it now contains TCGA as well as target data. Target is I like to think of it as a pediatric equivalent of TCGA. But very soon, there are going to be a number of other data sets there. Warren mentioned some of these this morning. Foundation Medicine, 18,000 cases. There are about 4,000 cases of other small cancer studies that are currently in dbGaP. The Multiple Myeloma Foundation and Genie, another 19,000. Warren mentioned those this morning. But in the next couple of years, we're also going to see data coming in from the NCI Match Precision Medicine trial, from the Clinical Trial Sequencing Program, Cancer Driver Discovery Program, a Human Cancer Models Initiative, and Apollo. And I'm going to talk about Apollo in a little bit later. But what you're seeing here is this is now approaching, we're over 76,000 cases, and we're really approaching critical mass of the amount of information that can really drive new discoveries. So the driver for the Genomic Data Commons was to unify some fragmented repositories. We had the raw data BAM files sitting in CG Hub being managed by UC Santa Cruz. We had derived data sitting at the NCI. All you could do was basically download these data and really not do much else with it. So the GDC was developed to really put in place a lot of a process around supporting the receipt, the quality control, integration, and storage and redistribution of these data sets. They go through an extensive process of harmonization of the raw sequence from both these legacy and new cancer research programs, and then applying state-of-the-art methods for generating derived data. And the important thing is that all new data coming into the system are processed exactly the same way. They're mapped to the latest version of the human genome exactly the same way. So this is really providing the foundation for identification of both high and low frequency cancer drivers, especially as the number of cases increases dramatically, defining the genomic determinants of response to therapy, and also for being able to select clinical trial cohorts that share genetic lesions. The second component of that we developed around the same time were the Cancer Genomics Cloud pilots. These were initiated 3 years ago based on our awareness that the traditional model of download and management by every researcher was no longer scalable. So we wanted to explore the effectiveness of co-locating data and compute capacity in cloud environments for access and analysis. And the overall goal was to provide access to large genomic data sets without the necessity of downloading, access to popular pipelines and visualization tools, the ability for researchers to bring their own tools and pipelines to the data, and also for researchers to be able to bring their own data and analyze it in context with the data that was already there in the database. And then finally providing workspaces for people to do their work in, to be able to save and share their data and the results of their analysis. All of this while meeting very strict federal security requirements. So, and Warren had this slide up this morning. We awarded 3 contracts to the Broad Institute, ISB, and Seven Bridges Genomics because we wanted to encourage innovation. They've each taken slightly different approaches to this problem. Broad and ISB have stood up in the Google Cloud. Broad has basically taken the Firehose pipeline and instantiated it in a cloud environment. ISB has leveraged the Google infrastructure to create novel query and visualization tools. And Seven Bridges has focused a lot on their existing platform and sharing interactive data exploration and more than 30 public pipelines. So in addition, so we have these 2 foundational pieces, but there now are other drivers for moving from where we are today to this more broad cancer research data commons. And I'm going to talk just about a couple of different programs that are beginning to generate large amounts of multimodal data. The Clinical Proteomic Tumor Analysis Consortium, this is CPTAC, was launched in 2011, and it really pioneered the integration of proteomics and genomics, looking at colorectal, breast, and ovarian cancer in its original incarnation, now identifying proteomic-centric subtypes, which turn out sometimes to be a little different than what you see in genomic data, and improving our understanding of cancer-relevant pathways through looking at post-translational modifications. The CPTAC is also collaborating with the Department of Defense and the VA in the Apollo program, Applied Proteogenomics Organizational Learning and Outcomes Network. So these are the two largest health systems in the U.S. And they're going to be analyzing DNA, RNA, protein expression, and medical imaging of 8,000 annotated human tissue specimens from a wide variety of organ sites. And then finally, again, back to the Blue Ribbon Panel, there are various programs that are going to be coming out of the Blue Ribbon Panel. As Warren mentioned this morning, a number have gone through our Board of Scientific Advisors and approval. Just a little teaser, one of these is going to be a human tumor atlas, which will generate a number of new data types, including the proteomic and genomic and cell imaging, but others as well. So today, we need to catch up the data from all of these. We've got the GDC for the genomic data. CPTAC has its data portal. And there's also the Cancer Imaging Archive, which has about 33,000 total subjects in the archive. But basically, we want to bring all of these data resources up to a more robust level to be able to manage all of these data. And that's where the Cancer Research Data Commons comes into play. So I want to show you graphically the state of the system today. So what I'm showing here is the current GDC and the cloud and the Cancer Cloud Pilots framework. As I said, the GDC supports the receipt, the QC, integration, storage, and redistribution of cancer genomic data sets. The Cloud Pilots complement the GDC by providing cost-effective, scalable computing resources for researchers to analyze these data. So researchers can access the GDC through a web interface or API. There are standard data submission processes for harmonizing the data, and users can search, visualize, and download the data directly from the GDC. Researchers can also access the Cloud Pilots through web interface or API. They're powered by commercial cloud providers, as I've said. They host the genomic data in the cloud directly. They also host analysis pipelines and visualization tools, and users can bring their own data to the Cloud Pilots. And both the GDC and the Cloud Pilots do authentication and authorization through ERA Commons and the dbGaP process for controlled access data. Here's where we see this in the near future. This is our new framework, where we're going to move towards a more streamlined infrastructure. We're calling the Data Commons Framework to enhance data sharing and big data computation for cancer research. So researchers will still submit data to the GDC. The data will be harmonized using their standard pipelines, and then those data will be mirrored in multiple commercial clouds as GDC nodes. The cloud resources will be able to access the genomic data from the commercial clouds as clients of these GDC nodes, and researchers will be able to access data and platform and perform analyses through the cloud resources. And also new algorithms, tools, pipelines, and visualizations are going to be available through users through various repositories. So just have DocStore here as an example. So it's a slight change, but essentially now you can consider the GDC footprint expanding from just the University of Chicago out into these commercial clouds. That's all part of the GDC as we know it. And then finally, our vision is to create this research data commons, where we're now managing more than just the genomic data, but initially genomic, proteomic, and imaging data. Data submission will be managed by subject matter experts in each of these fields, much as it is today. There are the equivalent of, say, data coordinating centers for proteomic data and for imaging data, and we're already looking at how we're going to manage the data from Apollo and the CPTAC program in exactly this way. Having the subject matter experts doing that initial QC and harmonization, and then depositing data in a centralized data store. The Commons itself will house multiple cloud-based nodes for these various data types, including not only genomics and clinical imaging and proteomics, but also cancer models, population data, functional clinical data, et cetera. And these will be interoperable through data standards and common identifiers. And then secure access to these control data will be provided to protect patient privacy. Don't take this slide as being literal, but we are, it's conceptual, but we are looking at a federated approach. This does not apply any single data store or any single platform. But the framework that we're developing right now between the GDC and the cloud pilots will form the foundation of this that can then be cloned and reused by these other nodes. To break this out, we really see these 5 components of a node. I talked a bit about that data acquisition and curation, where you have subject matter expertise focused on that, the people who know those data, the experts in that field, ensuring the quality and consistency of that data. There's infrastructure at the bottom, separate from that, which provides the physical hardware, the software, the security policies, the protocols, the cloud computing. There's support and help and operations of these systems to make this easy to use and maximize the impact of each of these nodes. And then there's an innovation component where new tools, new data types, mechanisms for researchers share the data, and that all of these things can be funded independently. For example, we have, Warren mentioned our ITCR grant program for informatics technology for cancer research would fund that innovation. Whereas there may be other funding mechanisms for these other components. And then finally, as part of one of our Moonshot activities, we're looking at a cancer data aggregator. You can think of this basically as middleware using standard APIs to easily aggregate data by case, by sample, by study, disease, tissue, et cetera, and providing that out through all the arrows are meant to show that there'll be many different ways to access these data depending on the type of question that you want to be asking. And then finally, I just want to acknowledge there are a lot of people who have been engaged in this. I just want to acknowledge my team who have been instrumental in the cloud pilots, Julie Clem and Tanya Davidson, and then the people here in the room, the ubiquitous Ishwar Chandramuleswaran, who used to work in my team before Maria stole him away to NIAID, and also here in the room, Ian Four and Azumi Hinkson, and a cast of thousands of others here as well. And with that, I will thank you very much. Okay.